Hello, everyone. My name is Sarah Alghamdi. I'm a PhD student from KAUST, King Abdullah University for Science and Technology. And today I will be presenting our work on answering this question. How much can model organisms' phenotypes teach us about human diseases? This is a study which utilizes ontologies and semantic machine learning. As you might know, model organisms are non-human organisms that are extensively studied in order to gain better knowledge of a variety of biological processes occurring in humans. More specifically, they have made significant contribution to the history of human genetic diseases research, both in terms of finding disease genes and explain their normal and pathological roles, meaning that if we want to understand the role of genes in diseases, then we need to know what they do normally and what happens when they are defected. A major challenge is to determine the gene functions. What is the normal role of gene and what is its role in diseases, if any? This means that functional genomics is critical to our understanding of the role of genes in diseases. We have functional annotations for much of the human genome. Have a look at Go annotations. But because some of genes have broad effect on phenotypes, then it's very hard to predict what is the phenotypes that might be associated with the mutation in a given gene. We also have information from for Mendelian diseases on mutant association phenotypes, but they're not complete and they don't cover the human genome. Additional annotation in model organisms derived from experiments or naturally occurring mutant in these species helps fill the genome-phenome annotation gaps in human. So we explore the model organisms' phenotypes to understand the potential contribution of human genes to diseases when we have no phenotypic data for from the human genes. Long ago, researchers thought about the integration of phenotypes for model organisms. And it has been studied using several approaches, using statistical approaches to study morphological integration, followed by the research on evolution and so on. Then this was put together systematically using ontologies, where phenotype ontologies were developed to capture and compare phenotypes within the context of a single species, with the formal class definitions that were utilized later to allow semantic integration, semantic data integration. The questions that we want to ask here in this research is the following. Can model organisms that are far away from humans still provide information about human diseases? We want to find out how much information from model organisms' phenotypes helps us to predict the gene disease associations, specifically for Mendelian diseases, only using the phenotype similarity over integrated phenotype ontologies to do this analysis without any actual biological knowledge. So we are not targeting specific pathways, only using phenotypic similarity. PhenomeNet and UPheno, the unified phenotype ontology, are two well-known ontologies which already have been developed for integrating and enabling the direct comparison of phenotypes between different species, both with the objective of allowing similar phenotypes from same or different organisms ontologies to be logically defined in similar forms. PhenomeNet ontology was developed by reformalizing phenotype groups and integrate them using parenthood relations. On the other hand, the UPheno ontology establishes some bridging axioms you can think about as generic classes that uses equivalence or subsumption axioms to connect phenotypes from different ontologies. Mainly, the difference between those two ontologies is in the pattern used to define the phenotype classes, which creates different classification hierarchies. And previously, we quantitatively showed how those different patterns can significantly give different performance in similar statistical analysis. The PhenomeNet ontology currently lacks classes for yeast and fly phenotypes, while those two classes are covered by UPheno. So we re-added fly and yeast phenotypes to PhenomeNet and used it for our analysis. So Pheno-E stands for phenomenon extension. And to incorporate fly and yeast phenotypes, we used similar strategies to Phenomenet, and we used fish and yeast phenotype ontology as well as Drosophila phenotype ontology. And we added some classes using the allele phenotypics informations from Flybase. As we said, the main goal in developing such ontologies is to allow similar phenotypes from same or different organisms ontologies to be logically defined in similar form. For phenotypes that are available in Drosophila phenotype ontology and fish and yeast ontology, we reformulate the phenotype class's description using the first formula here. And as Flybase has two types of abnormal phenotype classes, some associates allele with classes from Drosophila phenotype ontology, and some associate alleles with classes from the fly anatomy ontology, indicating that an anatomical or developmental structure was discovered to be abnormal in mutant fly. Therefore, we further defined new fly phenotype classes using annotations from Flybase. And to do so, we used this second formula. And as an example, this allele is found to cause some abnormality in the wing. So we defined a new class named phenotypic abnormality of wing. As you can see, those two formulas include Barthold relations, which is used for inference, constructing the hierarchy of the resulting ontology. This formulation allows us to form, allows classes from different resources to have direct subclass or equivalent class relations. As you can see in this example, here we color-coded the classes by their resources. Now we extended Pheno-E ontology. We can use it to answer our question. So we want to test the different organisms' contribution in predicting gene disease associations using only the phenotypic similarity. We predict genes to be associated with a disease if the phenotypic similarity between a disease and the gene's ortholog is high. So we used three different methods to determine the similarity between phenotypes of genes and diseases. The first method is semantic similarity using Resnick semantic similarity. Resnick similarity is an information content-based similarity measure, only uses the subsumption hierarchy. This method looks at the annotations of the classes and aggregates it in higher level, then calculate the information content. Then it determines the similarity between two classes as the information content of its most informative common ancestor. And do apply some averaging method to calculate the similarity between groups of phenotypes. But this method have one limitation, which is the fact that it only uses the subsumption hierarchy of the ontology, nothing else from the ontology. So to overcome this limitation, we applied embedding method. We applied OBA to BIC, which is an embedding method that learns the representation for ontology classes and annotations. This approach uses the asserted axioms and the inferred subclass axioms, and it also uses the ontology's metadata, labels, definitions, synonyms, and we can also add some annotations to the ontology and learn some representation for those annotations. But it has some limitations as it cannot explore more distant associations generated through the axioms. And to overcome this limitation, we further employed DL2BEC, which is another embedding method that expands the previous method. It converts the description logic axioms into a graph representation. So for example, a class description like this one can be divided into two edges, this one and this one, basically allowing direct edges represented represented for the axioms between involving two direct classes. And through the random walk, it can explore a chain of description logic axioms. And for the evaluation, we used unsupervised methods, basically following the illustration on the right. So as we said, higher phenotypic similarity between orthogonal genes and diseases indicate higher chance of genes to be associated with the diseases. And we used two evaluation data sets from MGI. The first one is the human evaluation data set, which associate human genes with Mendelian diseases. And the second one is mouse evaluation dataset, which associates mouse genes with mouse models of human diseases. And we evaluated the model organisms' phenotypes prediction using the area under the receiver operating characteristic curve. So the ROC AUC, and we used it in a micro method. So basically, we sort all the genes based on the similarity to each disease and then capture the true positive rate and the false positive rate at each prediction rank. And then we got the ROC curves and calculate the area under the ROC curves. We tested the phenotypes from mouse, from zebrafish, from fly, from yeast, and we also combined those phenotypes to annotate the human diseases. Here's one of the results that we have. And to allow direct comparison between model organisms' contribution in predicting gene disease association, we tested a very small data set where we used diseases that are associated with human genes, which have orthologs in all the four organisms, so in mouse, fish, fly, and yeast. And those orthologs have to have at least one phenotype. A total of 255 genes were used here. And we can see that mouse phenotypes are predicting higher ROC AUC using Semantic Similarity. But in the embedding-based methods, the combined methods, where we annotated human genes with phenotypes from all the organisms, produce higher ROC AUC results. Yet this is a very small data set, including only 255 genes, which suffices our conditions regarding the orthologs and phenotypes from all the organisms. A larger scale test is shown here. We cannot compare the results between the different organisms as it uses different number of genes and diseases. But what we can get from here is the big picture about what we can learn from. So basically, we can learn very well from the mouse phenotypes using all the methods, Semantic Similarity, DL2-Vec, and OBA2-Vec similarity in both data sets, and not so much from the rest of the organisms. And in some cases, you can see that even the performance is lower than expected when similarity is determined randomly. The combined model where we annotated the human genes with the orthologs from all the orthologs phenotypes from all the organisms seems to work using DL2-Vec similarity. Okay, how about this? Do we make better predictions when we combine mouse phenotypes with other organisms' phenotypes? In this test, we tested the bearing of mouse phenotypes with the rest of organisms following the same conditions as before. So genes have to have orthologs in both organisms and at least one phenotype from each of the organisms. And so here we have larger number of genes, and we can see that Semantic Similarity and DL2-Vec similarity are similar for the mouse in both methods. They show no increased signal when adding other organisms. Slightly higher AUC when combined with yeast phenotypes using DL2-Vec method. Finally, we compare our extension of PhenomeNet to UPheno. As for the comparison, we can directly compare the phenotypes from the mouse and the yeast phenotypes as both uses the same number of classes, the same classes IDs in both ontologies. And we can see here that both ontologies have similar performance in all of the tests. As a summary, we extended PhenoNet to include fish and yeast and Drosophila phenotypes. We applied semantic similarity and ontology embeddings in an unsupervised way to learn similarity between phenotypes. And we demonstrate that most of what we learn about human diseases from model organisms' phenotypes comes from the mouse data, not from the other organisms. At the end, I would like to thank everyone who helped and support this work. Thanks for our collaborator from Cambridge University, Professor Paul Schofield, and to my advisor, Professor Robert Hohendorf, and all the Bio-Ontology Research Group in KAUST. And thank you so much for listening. Please do not hesitate to ask any question.